Everybody, thank you so much for joining us today. Welcome to the RStudio Enterprise Community Meetup. I'm Rachel Dempsey. I'm calling in from Boston today. As you can see, we're doing things a little bit different from normal meetups today and are streaming out to LinkedIn, Facebook, YouTube, and Twitter. A huge thank you to Tom Mock, who's hanging out and helping behind the scenes as well. I'm also joined by my colleague today, Phil Bauscher.
Hey, everybody. Thanks for coming. I'm Phil Bauscher, the Director of Life Sciences and Healthcare here at RStudio.
If you've just joined now, feel free to also introduce yourselves through the chat window of wherever you're watching from and say hello, maybe where you're calling in from. But for anybody who's joining this group for the first time, this is a friendly and open meetup environment for teams to share the work they're doing within their organizations, teach lessons learned, network with each other, and really just allow us all to learn from each other. So thank you all for always making this such a welcoming community. We really want to create spaces where everybody can participate and we can hear from everyone. So I like to reiterate that we love to hear from everyone, no matter your level of experience or industry that you work in as well. While you can ask questions on whichever platform you're watching from, you can also ask questions anonymously as well through the Slido that we have, which I'll put up right here on the screen. But we are so excited to have so many members of the pharma community here today as well. So I'd love to use this opportunity to also share some information about a few other pharma communities and hand it over to Phil Bauscher from our team, who is the best resource for all of that.
Awesome. Thank you, Rachel. Very excited to be here today. So this is the 6th installment of our Life Sciences series. Previously, we've highlighted webinars by GSK, by Novartis, Roche, and Genentech, by Johnson & Johnson, and even the EPA. And you can find these webinars on our Life Sciences Champion page, which Rachel is going to highlight now on the screen. Over the last 7 years at RStudio, my team and I have helped many organizations use R for submissions, especially in the PK/PD space. And I would say in the last 2 to 3 years, there's been a lot of interest to use R for late stage clinical trials. And many of the organizations that we work with, they have different approaches to validation, to qualification, for risk, for reporting. And you can find some of our supporting documents that we have on our pharma page that Rachel is showing here. At the R and Pharma gathering that happens every year, you can see many of talks from the past conferences that highlight different approaches to submissions and how organizations are handling some of these things. I'll point out that the R and Pharma conference is coming up. It's a free virtual conference, will be in November, and we hope to highlight both how small and large organizations are approaching clinical submissions using R. And today we're going to be featuring one way that an organization has approached this, which is through Merck. And this is really going to highlight the awesome work from Nan Zhao, Yilong, our speaker, Kevin Anderson, Sarah Wang, Semya Yi, and countless others, people at Merck that have helped to enable the R reporting as well as documenting it for the community. It's just really fantastic. One example of this, which is very exciting, is the use of the R package GS design Kevin Anderson leads up, that was used for the Moderna vaccine. And so at R and Pharma last year, this webinar that we're featuring for you today was one of the most highly attended, booked up webinars or workshops that we had. And it's so exciting that we get to bring this for the community today and bring it to a larger audience and highlight this fantastic work from Merck. And so it's with a great pleasure that I'll pass it over now to Yilong to highlight and talk about this work that they're doing.
And I'll just also say, as mentioned earlier, we will have time for Q&A as well. So you can put your questions into wherever you're watching from, or use the Slido link, which I'll pull up once again right here. But I'm so excited to be joined by Yilong Zhang, now from the Health Technology Research Team at Meta, to talk about his experience at Merck, where he focused on developing clinical workflows and clinical reporting, and has been an amazing addition to the community. So with all of that, thank you all again for joining us. It's amazing to see such a great group of people here, and I would love to turn it over to you, Yilong. And we will go backstage. I'll bring you on here.
Yeah, thank you, Rachel, for that setup. Thank you, Phil, for the great introductory. Yeah, today I would like to share our experience of using R for clinical study report and submission. And this is a work where I'm at Merck to work with a group of talented members within and of course Pharma to share how we are thinking of the recommendation and experience for late-stage clinical trial to submit the work to the FDA or for the other regulatory agencies through the so-called ECD package to their portal. And before start, it's just a general disclaimer. So all opinion expressed here is just for my own and doesn't represent any organization. And some of the slides here, once you get the link, you probably can scroll down to see some full content. So before we start, I think it's just to reinforce the clarification from the FDA, right? From the FDA's statistical software clarification statement, it's clear to see that FDA doesn't require use of any specific software for the statistical analysis for clinical trials. But there's definitely a requirement to fulfill the 21 CFR Part 11 to complete the qualification of the software and also to conduct analysis in a GXP manner. So for here, it's more like we share how we are thinking to execute following this clarification statement to meet the requirement from regulatory agency to complete the analysis and also to generate a report that's ready for the submission. So with regard to the motivation, why we need to use R or different R package for regulatory development, right? So for late stage, it's also have many of the space that have a lot of the nice package to be used for the clinical trial development. For example, R has been widely used for clinical trial study design, for example, for group sequential design. As Phil mentioned, the GS design package for group sequential design has been widely used within Merck or beyond. And also recently, the development of non-proportional hazard group sequential design is also under development in R and handy and public available. And last year, Kevin just gave a nice seminar to the Deming conference to go through the information. And if you're interested, you can go to this bookdown project to see all the information. And secondly, so recently you can see a trend of using estimate framework following the ICH E9R1 addendum. So in response to that, novel missing data approach may need to be applied. For example, in recent paper, we collaborate with academia members for recurrent event data and robustness of missing data. Right? All those methods have been implemented as an R package and public available. So when a clinical trial try to apply those novel methods to their trial, instead of to re-implement other software, maybe we can just apply those R package available to the clinical trial study with a proper manner that package has been qualified and tested before it can be used for the final results. And as you may aware, R is also widely used for Bayesian statistics with a popular package like STAN, right, to be able to handle the Bayesian modeling part for hierarchical modeling. And sometimes for the network meta-analysis in HTA space, the drug reimbursement analysis can also be applied in this framework. So in addition, R always have the strengths for the visualization part, right? So there are some dedicated work related to the pharma. For example, the safety graphics working group has created a nice shiny app to summarize a bunch of quite commonly used safety graphs. And also another example, like we're exploring how we can use an interactive forest plot to help simplify the DMC safety monitoring review, right? So if you're interested, there's a recent talk by our colleague Yu Jie at the in Pharma conference to highlight how this small package Forestly can be used to create the interactive forest plot to aggregate the safety information and to have a quick review and digest by the DMC member. So these are some motivations that drive us to explore more of a proper and compliant way of using R for regulatory submission. So for some background, the work here, we're still trying to follow the ICH guidance, right? For the clinical study report, we're following the ICH E3 to structure the content of a clinical study report. And for the company book, r4csr.org, contain all the detailed information, step-by-step, how we can create some example tables using the tool we provided. And also we're trying to fill the gap to streamline the workflow, right? So in the same book, we try to discuss what's the concept for project management in clinical trial study space and provide some recommendations for that. And in the end, the most important part, once we complete the analysis in R, we need to find a way to properly submit the results and the source code and data to the regulatory agency through the format required by the regulatory agency. That's basically this eCTD format required by most of the global submission situation for drug development. And for the table, lists, and figures, for this talk, over focus still to deliver the results in RTF or Microsoft Word format, because in most of the pharma company, the RTF or Microsoft Word still play a central role to combine all the table, list, and figure into the clinical study reports. So to collaborate with clinicians and with the medical writing and the regulatory group, right? So people still using the Microsoft Word as a key component to combine all the things together. So that's how we play the focus on the Microsoft Word workflow, right? And it's also acknowledge that different organizations can have different way to complete their clinical study report, and the table standard can be different. So this is just one recommendation how we at Merck is handling those framework. And so for part that we work open space is about our consortium pilot submission, right? So our consortium frame a nice working group to organize people from both the regulatory agency and the pharma companies in different representatives to get everyone set together and also to think how we can submit the work complete in R to the FDA. So with this nice working group, so we contribute together totally in open space area. So you can go to the link like to identify who's a member and affiliations. And for now, what I'm trying to share is outstanding work by this working group for the first pilot that we are able to prepare the analysis package and prepare into the eCTD format and then submit to FDA with review. And here's just a short timeline, right? So about November last year, so we initiate this submission to FDA and then receive the FDA response in December. So in the response, we received some feedback and comments. So we try to address them and submit a revised submission to the FDA. And in March this year, we received the final FDA response. And all those submission and response letter are available on our consortium GitHub website that we will also have link in following slides. So if you're interested in the detail, how the package was organized and how those response communication between the working group and FDA. So all the information available on GitHub website, so you can review and explore. So while we are dealing with this first pilot submission, so few challenges that we need to solve. The first one is that every organization will develop some internal R package, for example, to streamline the workflow, to wrap up some open source R package available to a way that's easy to be create the table, figure, layout, following the internal standard. So we always need to have some internal code that need to be shared with regulatory agency. So one question is how we can submit those internal developed or appropriate package to the FDA, right? Because in the eCTD format, there are some restrictions that to submit, for example, we are not allowed to submit a zip file within the eCTD package, and all the file names should be in lower cases without any special character, right? And all the content should be in ASCII2 format. So how we're going to dealing with this with a simplified way? So that's first challenge we try to resolve. And basically a simple solution is to use a package called Patchlight. We will briefly introduce in following slides. And the second part is how we can follow the ICH or FDA guidance in preparing the eCTD package, right? eCTD have multiple modules, and typically we are following over content is in the module 5. That's basically the data set and the program for a clinical study report. And for the data part, typically we still follow in the standard requirement to use XPT format, and R also have a package to create those data sets. But for the first pilot, we try to focus on the analysis part by assuming all those XPT data set is already available. How we're able to submit the analysis program to the regulatory agency, right? And the last part, even though FDA didn't expect all the code is executable or reproducible from FDA review perspective, we still try to think how we can enhance the reproducibility from FDA reviewers perspective, right? How they can rerun all the analysis code we provided in the eCTD package in an easier manner, right? So what kind of step-by-step instruction we need to provide into the analysis data review guide. So those are the challenge we are trying to resolve in the first pilot submission with some simple analysis. And just highlight again that the assumption here is still about the analysis and the reporting part for the first pilot. We are not yet to touch the ADAM dataset or SDTM dataset generation to support analysis yet. But that can potentially be some future work for future pilot work. Right. So once we complete the first pilot, there are a few deliverables we combined into the eCTD submission package. So the first one, we're able to create a pseudo proprietary R package. I call it pseudo because it's also available on GitHub. So you can check what the package looks like. And also, we provide R scripts for the analysis. That's also on GitHub repo. And then, as part of the eCTD package, we also need to provide an analysis data review guide to detail, like, for example, what's a version of R and R package, and what's a step-by-step instruction to rerun the code, and also where to find the data set and output into the eCTD package. So all those information will be saved into the review guide and also be available on the eCTD package. So in reproduce the work, there are two open source GitHub repo. So the first one is more from development perspective, like as an organization, we need a space to save all the source code documentation, right? So that's all saved in this development repo to represent as an organization how we can organize all those information together. And the current recommendation for the first pilot is using an R package folder structure to organize all the information as needed. And those details also be discussed in the R4CSR book. And the second part, once an organization complete the development, then we need to reorg the information for the necessary part to be put into the eCTD package, into the module 5 of the submission package specifically. So in that part, this is a second repo called eCTD package repo that basically counts only the eCTD part that required to be submitted through the FDA portal to the agency, right? And once we complete the eCTD submission, so our consortium provides a nice announcement to summarize the achievement of this work, and probably you hear also from the LinkedIn or from the other channel to see how this announcement happens, right? So we also provide link here just in case you want to check the details. And after we submit the ECTD package, the initial one, to the FDA, so this is some highlight of the response from the FDA. So first, FDA reviewer confirmed that by using the R version 4.1.1, FDA was able to run the submitted code and confirm the applicant table and the submit figure in report TLF, basically the PDF file combined table, is reproducible. And also, FDA reviewer developed their own code to cross-check the results, right? So the analyst was able to independently generate all those tables using the submitted data. And there are also some minor comments being identified by FDA reviewer that we basically try to address in the revised submission. And if you are interested in those minor comments or suggestions, you can take a look at the initial submission FDA response. That's a full FDA letters to document the details. And for the revised submission, we basically to address those comments identified in the first submission. And once we receive a response, basically FDA agrees that the initial phase of this first pilot submission has been completed. And if you're interested on the revised one, you can also go to the second link here to see the results. And once we complete the first pilot, the working group team members start to brainstorm what can we provide more to the community, what would be over future work for the second or potentially third pilot submission. So there are a few things being identified and team members are working on those efforts. For example, we want to think if it's possible to submit a Shiny app within the ECTD package to the FDA, so the FDA reviewer can rerun the Shiny app in their local machine and to start to explore and digest information more efficiently in an interactive way, right? And second part, so the first pilot only targeted on FDA, but for an organization, typically we are thinking about global submission for drug development, right? So we also try to identify the potential point of contact for different regulatory agencies, for example, in EU, in China, in Japan, and see if we can also start the first pilot to those regulatory agencies and hear feedback to continue improve the workflow to potentially prepare a full submission using R for a global regulatory submission. And in addition, for the first submission, the analysis is quite simple and like simply the baseline characteristic table, some AE summary table, or a simple ANCOVA analysis for efficacy, right? There could be more advanced analysis require some complicated R package with certain system dependency, right? So we'd like to also explore the possibility to the submission with some advanced analytical method, for example, for study design or for missing data and for Bayesian method as we discussed in the motivation of this work. So once we discuss some high-level information about the FDA pilot submission, so I would like to go a little bit deep on how we are thinking about the reproducibility and also based on the guidance from the FDA, some recommendations we can take from the pilot submission. So these diagrams borrow from Roger Peng's famous science paper in 2011 to discuss reproducibility in general, right? So this diagram basically show the concept between the full replication, which represent, we can fully represent the data set and analysis, right? Which is unlikely to happen in clinical trial space because how expensive a clinical trial could be, right? So most likely, over reproducibility is more referring to the code part, right? With a given data set generated from a clinical trial. So how we can reproduce the analysis from the data set. So that's basically the part from the code side. And within the code side, there's also multiple levels about the reproducibility. So for the first part, right, so if we have some internal developed R package, then we need to version it and we need to reproduce with a certain version, right? And second part, there's also open space R version R package version need to be managed, so ensure the reproducibility. And in addition, sometimes you may see some discrepancy for the results between operation system. This is also true for many other software, right? So the assumption is that within an operation system, then we reproduce results. I think that's also the concept, like why we need a container for many of the analysis to ensure reproducibility, right? And one more even more challenging situation is that in very rare cases, you may get some trouble with different hardware, even though you are running the same operation system or you are running the same R and R package version. I would say this part is really unlikely to happen, but I just want to highlight this is also the part of need to be considered if we are doing something seriously. But for now, overall reproducibility is only focused on the first two parts. It's about the R version and about the R package version within a predefined operation system and hardware. Right. So in that case, this is how we are discuss the reproducibility. And with this given definition, so we start to review the requirement from FDA, like how FDA was thinking about the reproducibility of the code. So from FDA Study Data Technical Conformance Guide, we would just quote this paragraph. Basically, from the sponsor perspective, we need to provide a software program. We need to provide the ADAM data set used to generate the table and figure associated with the primary and the secondary efficacy analysis. In other words, the safety analysis code typically considered as optional, right? So the focus from FDA perspective is to run the source code for the primary and secondary efficacy analysis. And the second part, the specific software utilized should be specified in the analysis data review guide, like which R version, which R package version need to be documented properly to ensure the reproducibility. And also, FDA try to highlight that is to understand the process by which the variable for the respective analysis were created and to confirm the analysis algorithm. Basically, over understanding is that FDA, I see some background. Did I say recording?
It still sounds good to me.
Oh,
I can still hear you. Yep.
Okay, yeah, I just get my own voice. Okay, probably let's continue. So in that case of understanding that, so FDA didn't expect the code can directly be executable from their end. And the goal to have those software programs is to understand which variable in the ADAM data set has been used for the analysis and which exact analysis algorithm, for example, for ANCOVA model, what's your model terms, what's your missing data strategy to handle them in the code space, right? Because that's exactly what you do in the analysis. And also, FDA expect the software program should be in ASC-to-text format. In other words, those special characters shouldn't be put in the source code. So to achieve that, here's some recommendations from OVN. So first, when we are doing the final analysis for the clinical study report, we need to fix the R version, for example, R4.1.10. And also we need to fix a snapshot date, for example, August 31st, 2021. The purpose to have a snapshot date is to fix the RPackage version we are using for the analysis. And this is both achievable by the RStudio public package manager and also for the current time machine created by the MRAM. Or if within your organization, you have the commercial version of the RStudio package manager, you can have the time machine to both froze the internal and the external developed R package. Right. So this is how a snapshot date can be helpful to pre-specify the R package version. In the final analysis, you still need to specify each version of the R package used for your analysis. Okay. And also, we need to consider flexibility of input and output of tests, because when you code send to the FDA review part, the source code tests for the data set will be different. So some guidance to update the tests of the input and output should be provided in the analysis review data guide. And also, that's a part should be clarified into the ADRG. And based on our understanding, typically the FDA reviewer is doing the analysis in the Windows machine. And for some organization, maybe we complete our analysis in a different operation system, for example, Linux server. So in that case, we want to rerun the ECTD package and a dry run in over Windows machine as well to ensure it's fully reproducible from the sponsor end before we do the submission, right? And all those steps can be details in the ADRG appendix. And in the first pilot, we provide an example of step-by-step instruction in that case. So here we go to a little more detail about the implementation part, how this was handled. So in general, we share the same philosophy as a heavily recommends free package, right? So we just quote here. So anything can be automated, should be automated. So we want to do as little as possible by hand. We want to do as much as possible with functions. That's why we're trying to create our packages to simplify the workflow. And also a goal for our developers to think more of what needs to be deliverable instead of to worry about what our package looks like, right? So we want to bundle them together and be available in open space. And here are a few tools we developed to simplify that. So the first part is how we can generate a production-ready table and figures in RTF format. So that's how we create a package called R2RTF to achieve that. And the second part is mention how to get all those internal R package available into the ECTD package. So we create a package called Patchlight to represent our package into the pure TXT format, and also can revert and also install back into the reviewer's machine. And the last piece still under internal validation is how we can create a portable R environment to help the reviewer or help the dry run to complete analysis. Because on your machine, you can have different R version, different R package into your user space, right? What if the submission package have different R and R package version, right? You want to have a self-contained folder to reproduce that. Once you complete that, you can just delete, and that doesn't have impact for your system. And all the details is available in our CSR book. So if you're interested, you can go there to find the detail. For now, I just gave some highlight. So for the first one is about R2RTF. It contains several functions to pipe together to generate the RTF format. So that basically assumes the data set is already available. For example, you can handle that in tidyverse or other package. Once the data set is available, R2RTF will format it into the RTF for the production. And it's also that the other R package available to do the same work. For example, Roche create R package R table to do the same for the execute format. And Autoros have a package for Pharma RTF to also create tables in RTF format. And there's also other general tools like GT or Xtable and other tools available. So, right. So for here, it's just for our organization try to use this package to generate the files, but it's not necessary to be the only solution. And the second part is about to combine all the internal R package together into one TXT file that be able to save into the ECTD package module file. So Patchlight is a tool to pack all package into the plain text. It's also grammar to specify which part of your internal package need to be packed. For example, you don't have to pack your testing code. You don't have to pass all the linear of a R package to your part, because the goal is to have the source code in the R folder to be executable from a reviewer part, right? Also, it creates a standard. The TXT file can just be opened and be reviewed, so you can still take a look what has been assembled into the final submission package, right? So this is just an example code. Once you provide the path to the package, we can collate them. We have a convenient function to indicate what typically we consider to be saved in the ECTTP package, right? And sometimes you want to specify more. For example, you have some additional material in the INST folder. You can also pack multiple R factors into one file. So you have two package internally developed and saved into one TXT file. And once the TXT file is available, people can just unpack in their local machine. And you can also specify install equal to true that automatically install into your system. And the last piece is about the portable R environment, right? So to rerun the analysis, you probably need your .R project file specified RStudio configuration, a .R profile, or .R environment for that, right? So to create the run environment. And that also being enabled in CleanSlate. In addition, we can install a specific version, for example, R4.1.1 to a project folder. We can specify the R tools, because some of our package need to be compiled. You still need R tools in that part. So all those information can be covered into CleanSlate in this pipeline, and you can specify what information you need in this portable environment. Once it's set up, it can be used for the dry run or for potential FDA reviewer to review the content to simplify multiple steps in the ADR. Okay, so as a summary, I will try to think as a user story, right? So for as statistician, we can use tidyverse, R2RTF, or other internal tool to define the mock table, to create a listing and figures for the statistical analysis of clinical trial. Because typically you have more than 100 tables and figures for efficacy and safety for drug or vaccine development. Right? So you also need a way to organize them efficiently. As a programmer, we also use tidyverse or RTF and other internal tools to develop or validate analysis results based on those mock tables. And as statistical programmer, we also need to use Patchlight and also other internal tools to prepare those internal developed package in a way that can be saved into ECTD submission package. And in the end, so as an internal, external reviewer, we use CleanSlate to reconstruct the portable environment to reproduce analysis results. Right? We try also to give recommendation details in our Alpha CSR book. So for the folder structure part, when you have, for example, more than 100 deliverables, how you organize that? So our suggestion is to use a package folder structure to ensure the consistency, the reproducibility, automation, and compliance. Because there's already a lot of tools around our package that allow you to do the analysis and do some compliance check. So it's just one way we're thinking our package structure can potentially be used to organize all those things together. And also for project management, so we also recommend people to think in an agile way. So set up for success, like work as a team, because you always have multiple function areas to work together for the trial. You also need to think to design clean code structure, right, to make sure you have proper documentation and relation between of the functions, so other people can understand when they translate from project to another. And also, it's necessary to set capability boundary, right? In that way, people can cross boundary to help, but also be clear the responsibility of each role. And also importantly, we would recommend people to contribute to the community. When you feel like a tool that doesn't relate to the business logic, that can be standalone app package, I would like to share to the open space. In that way, we can continue to grow and to find a consensus to improve the workflow across the farm. And with regard to software development life cycle, it should also follow several steps like planning, development, validation, operation part. So in each of the steps, you need to be probably handled for internal develop R package and also need to be handled to qualify the external R package. Right. So those information, for example, can be referred R validation hub, white paper, how organization can achieve that. And there are many cross industry collaborations. So the work here cannot be achieved without all those outstanding collaborations with people from across pharma. So for example, if your interest is to understand how to qualify external R package, I would suggest go to our Validation Hub website and potentially join the working group. And there are several presentations being handled previously to understand the workflow and tools available. And for this talk, we discuss the R-based submission pilot, eventually on more future work. So more than welcome to check the website and potentially join and contribute, right? And also our pharma conference just wonderful. It helps us a lot to organize together, to share the idea, information, to collaborate. And in addition, the Arkansas adopting seminar series try to share the information, including this Meetup. Right. So continue to share the knowledge across the different organizations is important to help to shape a better solution towards the future. Yeah. And for the future direction of the current work we have, so we still try to enhance the compliance, reproducibility, traceability, and automation. Right. We want to also consider how we can assemble the metadata around the analysis, as you probably hear like the effort from CD360. Right. So we provide some prototype. There's some package called Patchlight and Metadata.aed on the GitHub Merck website. So in that way, you can check how we are thinking, how metadata was flowed from the planning and analysis reporting in the clinical trial analysis report. Right. We also want to enable some advanced study design statistical methods and also introduce some interactive visualization in the report for the DMC. And also can be without backend server. Okay, that's basically conclude all my presentation today, and thank you for listening. And
Thank you so much, Yilong. You can't hear us clapping, but we're all clapping. Here, let me pull Phil back on stage too. Thank you so much for a great presentation. I can see there's a lot of questions coming in already, so we'll do our best to try and aggregate them from everywhere. But I can see one question that has been pinned to the chat from Minoj, and apologies if I'm pronouncing your name incorrectly. It is, would R replace SAS completely, or its usage to some extent?
yeah, we don't think like R will replace SAS. Yeah, it's more like we add a more additional tool to the arsenal of the clinical trial development. Right? Like within our organization, definitely we can have different tools, even like Python, Junior, potentially can be in the clinical trial development, but we need a way to consider what will be a compliant and proper process for the development.
Thank you. And I just want to remind people too that if you want to ask questions anonymously as well, you can use the Slido link that I just put up on the screen. But otherwise, we're gathering questions from YouTube, Facebook, live stream, and Twitter. But I see one question that's been upvoted on Slido from Kevin is, if you're not using containers, is RMF playing a role in documenting dependencies?
Yeah, because our package itself already have a well-defined dependent structure in the description file. Right? And you can also use some nice package like IINF to log the file package version as well. Right? And that's also actually why Patchlight will also be helpful, because you just assemble all the package within a TXT file, and you can also unpack them. So your R and R package version has been fixed in the reviewer side. So I would say a container is not necessary, but will be a good add. But one challenge from FDA perspective is how we can enable container environment from FDA perspective.
Awesome. Thank you. I want to turn it over to Phil to ask one of the questions that came in too.
It's a lot of good ones coming in, but Yilong, one I thought you could help with here is a question says, what's the difference between ECTD and SDTM formats? Is one preferred, and does your package only support one?
Okay, so there are two different concepts. So SDTM is a data standard. So that's basically what the source data should be organized when you connect from the CRF. And ECTD is more like a folder structure to organize a clinical study report, like all the files from the clinical implementation operation, your clinical study report, your data set. Right? So SDTM data set, just one component stay in the ECTD package.
Thank you. I see. Okay, another upvoted question that came in anonymously that I want to be sure to ask is, I'm a grad student with limited R experience and interested in trials. The decision to make R my main software is a big one. Why would I want to make the switch?
Sorry, switch between what?
So I think they're talking about their decision to make R their main tool. What are some of the reasons they may want to make that switch?
I'm trying to understand. So definitely people have their own comfortable zone. They have their favorite tools for development. But as an organization, they have their own process, their standard. A group of people share the same consensus, how we complete the work. When people join an organization, I would suggest to first follow their existing process and consensus, and also to see if that match with your own interest and your comfort zone. Right. So I wouldn't say people need to change to use SAS or use R, but I would think it's more like a multi-language space. It's better to learn multiple language, to learn and borrow from the strengths of each language.
Fantastic, awesome. I think I can take the next one, Rachel, here. So to kind of switch up some of the topics a little bit, someone says for deep learning, Python is a popular choice. What efforts are made to address this in R? Yilong, has your team at Merck tackled any of the deep learning space for clinical?
Well, I have very limited experience on this, but for RStudio, I know you have some outstanding tool. Maybe you got better than
Yeah, so I can attach some links to a workshop that we teach, but there's been a lot of updates in this space. One is the fact that there's the reticulate package, which helps translate R to Python. It helps the interoperability between R and Python. So that's a big piece of it. You can also just come to Python directly, which is fantastic, and a lot of the ecosystem now supports that. There's also the TensorFlow package that's been created for R. JJ Allaire, our founder, gave a great keynote on this at the 2018 RStudio conference. So I'll put a link into the chat with some deep learning examples. Yeah, don't see a ton of this in clinical, but I was just curious, Yilong, if it had come up for you. So.
Mm-hmm.
I'm also going to share a link to a meetup that's coming up on June 1st on using Python in our RStudio team. If it's of interest to anyone too, I'll put it in the chat. But I see there's a question that came over from LinkedIn Live that says, "Thank you, Yilong, for the great talk. Have you and your team considered the use of R Markdown to integrate the report writing with the analysis and reporting?"
Yeah, the first pilot, R Markdown is used for the reporting part.
Awesome. Let me go back over to some of the LinkedIn questions here too. And one was, would FDA accept submissions with R despite the fact that R does not come with any warranty?
Well, so I wouldn't say it's not come with warranty. It's more like as an organization, you become a customer of an open source software, because that's not like a closed source, they have some pure statement. So our foundation also have a PDF file to clarify how R can be used in the clinical trial development part. And for an organization, it becomes the organization as a custom also to maintain documents to clarify as an organization how you are using open source software following the GXP guidance. Once that's been cleaned, I think that's sufficient for the analysis part. So as clarifying the FDA clarification, FDA say that you have to use some software that's being developed by a company and all closed source. Right.
Awesome. Thank you. Phil, I'm going to turn it over to you to ask the next question.
All right, let me see if I can find one over here streaming in from LinkedIn. All right.
Or I can ask one from Slido if you didn't find one.
So I found one here that I thought was pretty cool. It says, how are the formats handled in R? And I suspect that's a question on maybe the data formats for clinical trials. I know, Yilong, you're pretty active in that space. What was that like for you tackling the data management side of these clinical workflows?
Yes, the data format is language independent. So we definitely will follow the SDTM ADAM data standard. And FDA requires data to be submitted in XPT format. That's open source format, even though it's developed by SAS. So in other words, this is the same workflow we need to achieve, no matter we are using any software language.
Great. Thank you. So I'll go over to some of the anonymous questions. And one is, what steps did you take to validate the R packages themselves?
Yeah, I will just recommend one Pharma paper we developed last year. So we provide clear guidance, like a step-by-step instruction. So in general, it's follow how to recommend our package book, right? So we use a structure to go through them, but we need to pre-specify the testing plan. We also need some independent testing and potentially double programming for the code. So like as an organization, you definitely will need some execution resource to qualify and develop internal package. So this can mimic the existing process for other language you already have in your organization.
I think I got one here, Rachel. That's pretty good. Someone's asked about how to approach macros in R or similar things to macros in R. What has that been like for you using R?
Yeah, that's also a question discussed a lot in why we started R part. So for now, we're trying to think R to provide end-to-end solution, because the way to mix multiple language can create additional challenge from the IT, from your internal process, because which part of the process you want to use SAS R become unclear in your process. So for now, we haven't go through the detail how to hybridly use SAS and R in one particular analysis. But in a study, when you have multiple deliverable, right? You can have some deliverable in SAS, some deliverable in R that can be executed independently.
I think one thing too, the question's trying to tackle is what were similarities in the R experience for you in putting together the clinical trials to what others may have experienced with macros? Was that the functions that you built? Was it the packages that you designed? You know, did you have different routines or workflows that you shared within the team in R that were reproducible and could be packaged up and shared?
Yeah, my experience that different companies have different workflow for how to use SAS macro to generate the results. And it's the same in R, right? Probably if you consider Merck's flow, Roche's flow, that can be different, right? So in the R for CSR, we provide one recommendation how to do the project management, and with project management, where to save each of the files in a folder structure within our package. Right? This is how we are thinking a way to handle like a large amount of table figure listing required for analysis.
Thank
So
you. Oh, sorry,
yeah.
Phil. Did you want to continue on that one?
I was just going to ask a question that I'm selfishly interested in, just based on some of the work that we're doing with R and Pharma, is Shiny. Did your team look or think about using Shiny in the submissions process?
Well, this probably you can get more answers from the Roche team. Yeah. So definitely, I think the second pilot, as mentioned, is about the Shiny app part. I think the team is actively working on that to explore how to use Shiny with the ECTD and potentially reproduce in the review side.
Very cool.
Thank you. I see Sylvia asked a great question. I think it was coming through from LinkedIn. And it was, thanks, Yilong. In your experience, are the external reviewers at FDA for R versus SAS the same group? Trying to think if there would be a delay caused in reviews since SAS is more popular for the past 2 decades.
So I wouldn't represent FDA. My general hearing when we have the working group, so FDA member have people familiar both R or SAS or either of them. And also in FDA's guidance, it's also recommend you indicate the software you used in the Type C meeting for the pre-IND submission. So in that way, FDA can be aware what kind of software you are trying to use.
Sorry, I froze for a second, Phil, if you can take the next question.
Oh, yeah. So I see, let me see what we have here. The page is refreshing on me, so I'm trying to keep up with how fast it's moving. So one question that I saw was about Admiral. I don't know where it went, but I think the question was, people are interested in using Admiral for prepping data sets for clinical trials. Here I just found it. It said any other package for clinical trials data set creation which is useful. I don't know if we lost.
Yeah, I think Merck is a wonderful project. Another group of people are trying to see how to develop the Adam data set code in a central place. Right? In general, for most data manipulation, I would say type diverse is sufficient. Right? So if for your exploration or for develop our code for the data set generation.
Thanks, Yilong. I see someone else asked on LinkedIn, is the Shiny Meta package a better way to build reproducibility for the FDA?
I don't have expertise on that.
So I can just provide some color. So Shiny Meta is a package that was created by the Shiny team in RStudio probably about 2 or 3 years ago, and it helps implement reproducibility within workflows within Shiny applications. So I would say, stay tuned or watch some of the Shiny for Submissions working group information on the R and Pharma website. There's a couple different approaches to reproducibility via Shiny, and it will be interesting to see, what becomes more of a standard as more Shiny apps are shipped off through the submissions process. But I do think it's a great way for reproducibility, but it also takes a little bit of learning. So it will be interesting to see if it's adopted or if there's other preferred approaches.
Awesome. I just want to take a second to address a few questions about where the slides exist. So I just put on the screen right here the slides for today that Yilong had shared with us before the event. And then also a few questions around the recording. So one of the great things about using this live streaming tool is that the recording will be there immediately on YouTube. So I'll be sure to share that link there as well. Okay, I'm gonna go over to a few other questions, and thank you, Tom, for helping organize these for us in the back end. Okay, this is a bit of a long question, so let me take a second here. Tanisha asked, what is the expectation? So, oh, sorry, it disappeared for a second. Okay, all data sets and display creation in SAS and QC in R, or vice versa, or only display creation in SAS and QC in R. And I think everybody can read the, the question too, but, or a total mix and match of SAS and R would be fine for FDA submission.
Yeah, the voice have some lag. If my understanding correct, you are referring some data set created in R or validated in SAS or vice versa. So what's the question?
Can you see the question on the screen too?
Somehow
Long
no.
question. Okay.
I think what they're getting at is, will submissions be a mix and match of SAS and R? Because it's quite a big question. Maybe just comment about that, Yilong. What do you think organizations will mix the two? Will organizations choose to use R? Will they use R internally for the prep? What do you think? Or what was this like for your team?
I just feel it's a multi-linguistic way, right? But for particular task, it's better to be consensus on the tool to be used, right?
Yeah.
For example, if the goal is to create an analysis for survival, it's better all the trial for organization is using the same way instead of you have multiple way. Even within SAS or within R, you want to use only one R package instead of multiple packages to do the same thing.
Thank you, Yilong. There are quite a few other anonymous questions, so I want to make sure that I get over to those as well. And just so everyone knows, we'll be collecting all the questions and make sure that we answer these too. So maybe in some blog post format as well. But one of the questions is around, so it says, are there any security issues with using R versus Python? This person said, my hospital IT team is less familiar with R and has questions about how secure the packages are.
Phil, would you want to address that?
I mean, I think the thing to know, and I think is important, is that CRAN and the R Foundation does a wonderful job of running checks when packages are submitted, and that typically is a pretty good thing to lean on and to understand. And some organizations have some scanning tools and things that they use internally. But that usually is enough for people to understand for what it takes to get a package to CRAN, which is not, it's a big undertaking and it's a lot, and for package developers to maintain and support. And so that's a big part of it.
Awesome. I just put a link on the screen right now that might be helpful for the person who asked the question too. So it's just our champion site. And there's a separate section on working with IT too. Phil, do you see any other questions?
I think we're after the time. I don't if we can keep going all afternoon if that's okay, but I'm sure Yilong's got to get back to his work, but I do see some more, maybe a blog post and aggregating and collecting these up would be the right way to approach it, because Yilong, there's a lot of interest in this. There's a lot of questions coming in for you too. So,
cool.
Yeah. Rachel, how far after did you want to go today?
Well, I don't want to take too much of Yilong's time, because thank you so much for the awesome presentation too. Do you have time for a few more?
Yeah, probably. How about 2 more?
Okay.
2 more. All right. You pick one and I'll pick one. How about that, Rachel?
Okay. You go first.
All right. Let me see here. Let me see what's coming in. Let me see. So I think the one that is interesting here would be, so I see a lot of questions, and I think about Docker and containers and using that infrastructure for some of this work. I'm interested, Yilong, what that was like. Did that was that part of the conversations? You know, what would that look like maybe in a hypothetical world with the FDA or other regulatory bodies? Has your team thought about that?
Yeah, personally, I haven't tried Docker. I basically have very limited experience on that. And it's definitely a bring up in many conversations for the submission working group. I think the one challenge is still how to get FDA ready to receive the information. But from your own organization, I think Docker could be a very good way or container solution. But I would think like Eric, Andrew, or Doc from different teams, they have better understanding or potentially a live stream on Docker itself. So that would be nice.
Great, thank you. So one other question, I'll go over to the Slido. There was an anonymous question, which I know we've touched on reproducibility a bit, but what's the best practice for a group of programmers using R for submission to avoid any challenges with reproducibility?
Oops. Yeah, sorry, I still hear the lag of the voice.
Sorry, what's the best practice for a group of programmers using R to avoid any challenges with reproducibility?
Well, I think first thing, start to use version control. Second, have your R profile set properly and in a shared space that people can all access the same location. Right? So I think in general, that would be some good way to enhance reproducibility. And if you're familiar with RIn or other package, try to use them to log your R package version. Right?
Awesome. Well, with everybody here, I do want to also take just a second to let you know that we do have another Pharma meetup coming up in August. And if Tom can help me behind the scenes, I tried to prepare a caption for that to pop up. But if you want to just see all future events, you can use the short link we just put on the screen, rstd.io/community-events. But we will have a Pharma meetup specifically on August 23rd, led by Afshin at Bristol Myers Squibb. And Afshin will be presenting on data as a product, so data science framework for data collaborations. And I think that will be a great event as well. I'll share in the chat just the add to calendar link if you want to just add that right now. I'll put it right here. There you go. And I'll show that on the screen. So just rstd.io/pharma-meetup, and you can add that event to your calendar too. But thank you so much, Yilong, for sharing your time with us and your experience. It's great to be able to learn from you and to see all these awesome questions from the community too. Phil, I'll let you weigh in as well.
No, thank you. It's always been exciting to see the work that you do with your team. I always love following it and watching it, and yeah, excited to see what you still produce.
Cool.
Thank you.
Thank you, Rachel.
Thanks. Yeah. See ya.
Have a great rest of the day, everybody. Thank